So she did her postdoc at the Rockefeller University in New York and identified novel mechanisms involved in the Joubert syndrome, which is classified as a ciliopathy. And her current research at the Department of Human Genetics focused on the molecular genetic elucidation of inherited retinal diseases and trying to solve the unsolved cases with IRDs. So today, Susanne will talk about the complexity of IRDs, clinical and genetic heterogeneity. Susanne, thank you very much for accepting our invitation, and we look forward to your presentation. The screen is yours.
Thank you for your nice introduction, Aurélie. Let me share my screen. Let's put myself on the side. Yes. Ooh.
Yeah, it's fine.
Yeah. So completely different topic than this morning. We're going to talk about the complexity of inherited retinal diseases, and there will be no fundus images in my presentation. So a warning on that. So I want to tell you a little bit more about the, I have my bar here. Can I remove that? No, well, I'll try. So all the clinical phenotypes that you heard about this morning is a wide range between rods and cones and severity. And what we know of that there's already more than 280 genes that cause these different type of clinical phenotypes. And one gene may cause different phenotypes, and one phenotype may be explained by different genes. And so for all the patients with an inherited retinal disease in a clinical diagnostic setting, we are currently still using whole exome sequencing. But 40% of all cases are still left genetically unexplained. And whole exome sequencing has its limitations that it cannot detect coding variants, structural variants are missed, there may be an incomplete coverage, and we may just not know enough about variants that may be causing after all. So at the bottom here, you can see what a whole exome sequencing covers. So it really covers the exons only. So we will detect missense, nonsense, frame shift, and canonical variants. So what we hope to detect is a different type of variants that are called splice site variants. So on the left side, I'm showing you again what an exon is. And in the box over here, these are called the canonical splice site variants. So these are 100% conserved for each exon. And a variant in these will definitely lead to exon skipping. Then we have the non-canonical splice site regions, which are the regions surrounding these canonical splice site variants. And also their variants can affect the splicing of an exon that may lead to a clinical phenotype in the end. Then we also have deep intronic variants. They are beyond the non-canonical splice site regions outside of the exons. And these variants may lead to alternative cryptic splice sites and thereby pseudoexons. And these variants together, these three, the last ones that I mentioned, these could be a specific target for variant-specific genetic therapies. And I think you will hear more about that later today. And then there's also structural variants, and I will not go too much into detail about these today, and also not the one that Julio mentioned earlier, and I will only briefly touch upon them at the end. So in a recent study, we are doing whole genome sequencing in 103 currently unexplained IRD cases. So this does not only cover these exonic regions, but also has coverage in the entire genome. So that will help us to also find these variants that are in these regions near an exon, or deep intronic variants that may lead to a pseudoexon. And from these 103 patients that we selected, there were 45 Dutch patients, 19 Israeli, and 39 Irish patients. And we all had them as a suspected recessive inheritance pattern. And these studies were done by Zainab Fadaya and Laura Whelan from Ireland as a guest PhD student in our lab. And these patients were all tested with either whole exome sequencing in a diagnostic setting or with a panel with the currently known genes at the time. So how do we select these variants and where do we look for? So if we only know already one variant for the two, if it's a recessive disease, we look at genotype-phenotype correlation, because that will be the most obvious candidates. We look at rare variants. So we use GNOME AD, and that's a population database with all allele frequencies. And we're looking for something rare that should not be common in everyone. Then we make use of Alamut Alamut prediction scores and Splice AI. They are programs that can help us to determine if a variant would be possibly pathogenic or not, or have an influence on splicing. And we also try to look at splice site silencers or enhancers that may give us a better view on if a variant may or may not have an influence on splicing. For the missense variants that come from the 3.6 billion variants for an individual patient, we of course already look at the frame shifts and the stop mutations. They are relatively obvious, but then for missense variants, we only want to look at the strong missense variants. And for that, we use the PhyloP, the CAT-FRET score, and the Grantham score. And they say something about the nucleotide conservation, if a variant may or may not be deleterious, or if an amino acid change is very strong. And if in our lab, we use the criteria, if a variant meets the 3 criteria, then that will be a strong indication to look at that variant. If a variant has less of these criteria, then we will look later at these. And for the splice site variant, we, as already mentioned, we use Alamut and Splice AI. There we have made the internal rules that if 2 out of the 5 programs show a 2% increase, then we think that that may be suitable to look into it, and perhaps that variant has an effect. And for Splice AI, which is a little bit newer, we use a 10% cutoff. So if we have variants that are candidates and we think that they may have an effect on splicing, we're going to test that in an in vitro system. For that, we make a construct with a large genomic region surrounding the variant of interest. And we make a construct in a mutant and another one with a wild type version. And we make that into a construct with the rhodopsin exons as they are retinal retinal genes. We transfect these in HEC292T cells, and after 24 hours, we can collect the RNA and do a readout on electrophoresis gels. So there we can see what the result is on the wild type versus the mutant. And with that RNA analysis, we can detect events like exon skipping, exon elongation, exon reduction, or pseudoexon inclusion. Or perhaps there's no difference, and then the result will remain the same as the wild type. So this is in one view, the result that we have found from the 103 genomes. And as you can see, I'm showing 100 because one part was taken out for a different manuscript. So, but from these 100 probands, we were able to solve 24%. And you may think that's not a lot, but these probands were all strongly pre-screened already. So this is still a good result, I think. And on the right side, you can see that in the inner ring, I'm showing you the different phenotypes of all the patients that we have in our cohort. And then for each phenotype, I'm showing a different type of variants. So you see that there's not one clinical phenotype that always has the same type of variants. It's just a wide range. And here in a little bit more zoom in, I'm showing you a table with some of the results that we have found. So some of them have an obvious genotype-phenotype correlation. If you are familiar with the genes, for instance, Oguchi disease is, for me at least, it's a recognizable gene if I see a variant in GRM6. This variant was already found, and along with that, we found a variant that fits with the genotype-phenotype correlation, and therefore we can say that this patient is explained. But some of them are obvious genes, and they are not just missed by any reason, but often they were missed because the target or the panel that these patients were screened with did just not include that gene yet. So I'm showing you 4 examples today of variants that we found in our whole genome sequencing efforts. So this is a very, very deep intronic variant, 70 kb into the intron, so from the exon away. So on the top side, I'm showing you the wild-type genomic coordinates and the base pairs. And this is the mutant. And these are the scores for 2 of these Alamut prediction programs. Over here in green, I'm showing you an acceptor site with its corresponding scores, and the donor site depicted in blue with its predictive scores. So these are already relatively reasonable prediction scores if it's a range from 0 to 100. But because of this variant change from an A to a G, these scores go up quite a lot, so nearly to 100. So that's a strong prediction. So our prediction was that this variant will lead to a pseudoexon. And I'm showing you here the result of what we saw on the gel. So indeed, in the wild type, this is the wild type product. There's no wild type product in the mutant construct, but it does show this pseudoexon. And you do see it a little bit also in the wild type situation, which may be naturally occurring pseudoexon, and that's not strange if you look at these scores. But in the mutant, there's no wild type remaining. And if you sequence this, you definitely see this exactly these same boundaries as we predicted. So this patient for us is solved as it had another variant in ICE as well. A second example that I'm showing you, there were 4 patients that had as a first allele a variant in BBS1. And this is a known mild hypomorphic allele that is seen often with RP patients, retinitis pigmentosa. And for all these 4 patients, they were not related. We found the same variant at a -21 position. And this was a special variant because I'm showing you again here, the wild type on top and the mutant at the bottom part, with here the exon boundaries. There was quite a high score over here. But what we saw over here was a difference in a branch point. And that is something that we had not seen before. I'm showing you here how does this look in Alamut. Alamut shows you on the top also the wild type and at the bottom the mutant. And over here, this separate track shows you a score for a branch point variant, which is just not present anymore in the mutant. So we also thought that this was interesting to test because this A is exactly the most important A in a branch point motif. So we really thought that if this variant changes, there should be an effect on splicing. Because what does this branch point do? That is the A branch point position that is necessary for if the 5' splice site is cleaved, that will attach to that specific A. And that A is no longer there in the patient. So we assumed that exon skipping of 2 was going to happen. So we made that construct in a wild type and a mutant version with a lot of genomic context around it. And what we saw that was there was a product that was normal, going from exon 7, 6, 7, 8, and 9, a prominent product of exon 6, 7, partial skipping of exon 8 and going to 9, and 2 minor products where either exon 8 was skipped and exon 7 and 8 was skipped. So what we assumed that there would just be exon skipping. We noted that in Alamut also, again, there was an alternative branch point predicted. And therefore, we think that now an alternative splice site is used within the exon. And that was the prominent product. And this was the cause of the disease. And this was published in a separate paper. The third case why we think whole genome sequencing is very important is because of this case. In this patient, we did find, or genome diagnostics already found 2 variants in 2 different genes that are both known to cause Leber congenital amaurosis, the phenotype of this patient. So we obviously zoomed in on these genes to look for the second allele, but we did not find any second variant in the coding regions or the intronic regions. But we know a lot of genes that are known to cause LCA or related diseases. So we had to go beyond these genes. And what we found was a homozygous deletion in a third gene that was associated to LCA. So it was an exon spanning deletion that leads to an out of frame deletion. And the funny part about this is that the breakpoint of these two sites, let's say, they have microhomology. So these nucleotides, they can either be on this side of the deletion or on this side. And that is also probably why it happened. And this was not picked up in whole exome sequencing because that is told to only look at deletions if they are exons, if they are 2 exons or larger. And then the last example here is that we found a whole gene heterozygous deletion. I told you that we included only presumed recessive inheritance families, but this is a gene that is known to autosomal dominant retinitis pigmentosa, but it shows reduced penetrance. So in this case, we really have to go back to the family because likely either the mother or the father will carry or they have to carry the deletion, but they may or may not show a disease. Oop. So the take-home messages for today is that we did find the causative genetic event in 24% of all cases that we studied with whole genome sequencing. And I hope I convince you that we do find different type of variants and many genes, and that it's a really big search for these variants. The currently unresolved probands, they may have compound heterozygous or homozygous variants that are only in the intron, and we have not focused on these yet because that is simply a very challenging task. And as we only looked at cases that are currently known IRD associated genes, we cannot exclude, of course, that we will find genetic mutations in genes that we just don't know yet. So that is something for the future. And branch point variances are still a new type of variance that we have to look into. So that is also, we have learned from this process, and we will, of course, use that knowledge for future research. And I hope that I told you, shown you today that the functional analysis of these non-coding variants is important. I have to tell you that for some of the splice assays that we did not find an effect, while they had strong predictions. So we have to realize that we are using an in vitro system that may, in hex cells, that may not be this the best representation of what's happening in the retina. So for these type of variants, we would want to test photoreceptor progenitor cells, and that would probably be a better representation of what's really going on in the retina. And we have to continue these studies as it's becoming more and more important for patients to know their genetic mutation in order to be included for genetic studies in the therapeutic studies in the future. Current efforts are ongoing to study the unsolved patient with innovative approaches. I don't have too much time to go into detail for these today, but we're using optical genome mapping and long-read whole genome sequencing to find complex and large structural variants. And a recent publication that we have is on a hidden inverted duplication within the gene in a choroideremia family. And also we do need to study still the structural variants that lead to chromosomal architecture aberrations. And that's also something that I do not have time for today, but we have found recently that there was an ectopic expression event in the retina. So a gene that should not be present in the retina was turned on by mistake and led to autosomal dominant RP. So I would really like to thank you for listening and all the contributors to the study, with a special thanks to Zainab and Laura, who did a lot of work on this big study. And I want to thank you for your attention. And here are the QR codes of the 2 papers that I just mentioned, in case you're interested to know more. Thank you.
Thank you very much, Susanne. This was a very interesting talk. I forgot to mention to everyone that they can post their question in the chat, or they can just raise their hands. So I hope you have questions. I may be waiting for some of them. I can start. It's the privilege of the moderator.
Sure.
I was wondering, so you mentioned that some of the prediction did not translate into a phenotype in your in vitro model. Can you tell us approximately the proportion of the in silico prediction that translate into something and the one that did not work?
Well, that also depends a little bit. We have quite strict criteria, and you learn by doing, of course. So we do get better at making these predictions. I think for now, about half of the variants that we test in a MIDI gene assay are what we truly found. With the entrance of Splice AI, our prediction has become a lot better.
Okay.
So we now use Splice AI as a main lead. If Splice AI does not give a good prediction, then we may use alternatives. But there also we have to consider that Splice AI is a prediction program that is trained on different type of tissues, but not the retina.
Yeah, okay.
So that is also a study of, I think she's also here, Tabea Riepe. She's in a project working right now, so that we hope that we can improve this predictions of Splice AI by adding retina data to the training, let's say.
Okay, yeah, that's very interesting. Alex, you have a question?
Yes. So you mentioned all this new technology that you are using, optical mapping, and you're using also whole genome. So how close do you think we are to solve all this genetic complexity for IRDs? Are we still far, far away, or do you think that in a couple of years, all the unsolved cases might be solved?
That's a good question always. I think we're getting there, but we're getting to the low-hanging fruit is gone. And also with whole genome sequencing, we start with the obvious ones, the genotype, or the patients that have a very obvious phenotype or different than a retinitis pigmentosa can be explained by 80 genes. There are some phenotypes there that are explained by 1 or 2 genes. So those would be the preferred patients to start with if you start exploring a new technology. And that's also why with optical genome mapping, and we are already quite successful in finding new genetic events that the short read whole genome sequencing that I showed you today was unable to detect. So I think with every new technology, we will find a different type of events that were missed with a previous technology. And but I hope that in a few years we'll get there. I think that the number of genes is reaching a plateau, and it's just learning more about what's happening in the introns and structural variants.
Thank you.
Oh yes, sorry. Thank you very much, Susanne. If there's any more